We found that certain carbohydrate molecules play important roles in brain functions such as social memory. And we think this is exciting because it has implications for understanding how to repair neurons after injury and neurodegenerative and psychiatric disorders.
Hello, I'm Emily Schneider from the American Chemical Society, and welcome to this media briefing from ACS Fall 2023. Can you recognize someone you haven't seen in years, but forget what you had for breakfast yesterday? Our brains constantly rearrange their circuitry to remember familiar faces or learn new skills, but how this process works on a molecular level isn't well understood. Today, scientists report that sulfate groups on complex sugar molecules called glycosaminoglycans, or GAGs, affect our brain's plasticity. Determining their functions could help us understand how memory and learning work, and even provide ways to repair neural connectivity after injuries. We're joined today by Linda Shea Wilson from the California Institute of Technology. Thank you for joining us.
Thank you. It's a pleasure to be here.
How did you get the idea for this project?
I was trained as a chemist and as a neurobiologist, and so when I started my own lab, I was wanted to study molecules that played important roles in the nervous system. And the molecules that I kept reading about were carbohydrates known as glycosaminoglycans. Um, these molecules had been implicated in a wide range of functions, but their roles were not really well understood. And so we got excited about studying the molecules and using them to understand how the brain works on a molecular and cellular level.
So what is so interesting about these glycosaminoglycans, these GAGs?
These glycans are, I think they have really fascinating structures because they're long polymeric chains, so they can have up to 200 um, monosaccharides or sugar units. And then along the chain, sulfate groups are added to essentially decorate the chain. And you can decorate it in many different ways. So there are, thousands of potential structures that can be built through within this chain through the different sulfation patterns.
And what were you trying to find out with this research?
And we were trying to understand what these carbohydrates were doing in the brain. The molecules were known to undergo changes in their chemical structures during development and after neuronal injury, but it wasn't clear why. And so we wanted to understand why the molecules were changing in their structures and what effect that was having on the brain.
And how did you investigate this?
We used a combination of chemical and biological tools in order to tease out what the molecules were doing. Using chemistry, we could build the molecules, synthesize them from scratch, essentially, and test their effects on nerve cells. We also used biological approaches, such as genetic approaches, to perturb the structure of the molecule in cells and then looked at the effects. And so really the combination of chemistry and biology gave us insights into how the molecules were affecting neuroregeneration and brain function.
And what did you find out?
Well, we figured out that certain forms of the molecule were altered, altered the brain's plasticity, the ability of the brain to adapt and form new neural connections or strengthen existing connections. For example, we found that the sugars were decorated with sulfate groups. Their structures were being altered in response to neural injury. And that was enabling them to inhibit the regrowth of the cells. And then other forms of the molecule, different sulfation patterns, led to changes in the types of synapses or connections between nerve cells and affected social memory, the ability to recognize a familiar person or object.
And then what's the big significance of finding out how these patterns change and how that affects, neuroplasticity in a sense?
Well, I think they point to ways potentially to treat neuronal damage. So that's one of the things we're excited about. By blocking this inhibitory carbohydrate, we found that we could promote neuronal repair. And we think that understanding how these molecules affect social memory could give us insights into neurodevelopmental and neuropsychiatric disorders such as autism, schizophrenia, and bipolar disorder, which are often characterized by these, deficits in social memory and the types of changes in the synapses, the connections that we observe this molecule produces.
Were any of these results surprising?
We weren't expecting the effects on social memory because the molecules had never been linked to really any higher order cognitive functions or memory. And so we were really, excited to see that the molecules were affecting a particular region of the brain that was important for social memory, and then following up on those studies to discover that they were actually affecting memory itself.
Could this ever see any clinical applications?
Yeah, I think there are. We're really not at the stage directly, right, where we are working on the clinical applications, but I think the implications that we would like to pursue in the future would be the potential to repair axons after neuronal damage in the case of injuries like stroke or spinal cord injury or even neurodegenerative diseases. And then, in terms of the social memory, to see whether we understand that process enough to be able to think of, devise potential treatments or targets that would be relevant for those conditions.
So what do you really want viewers to take away after they listen to your talk?
Well, I think carbohydrates are not often thought of as, molecules that are important for brain functions. And so I think that's one of the key takeaways. They can have profound effects on neuronal functions, and many of the roles are still left to be understood and studied.
What's the big next step?
Well, I think, we'd like to, of course, understand even more deeply how these molecules are affecting those functions. They're very complicated functions that we're trying to learn about on a molecular level. And we'd like to understand or develop kind of new ways to modulate those functions. We may be able to manipulate the structure of the molecules to affect axon regeneration and synapse formation and social memory by controlling or tinkering with the structure of those molecules.
Why is this research so important?
So most people think about the brain as being a collection of cells, right? We have 100 billion neurons that talk to one another. What we found is that the molecules that are present between the cells, which is carbohydrates and other molecules, play a really important role in mediating that communication between neurons.
Well, thank you so much, Linda, for sharing your research with us.
Thank you. It's a pleasure to be here.
Be sure to check out our other media briefings for ACS Fall 2023, which will be posted throughout the meeting at www.acs.org/ACS Fall 2023 briefings.